Hello students, welcome back to Beat the Basics. In this video, we will be talking about another cell organelle, endoplasmic reticulum, or in short, ER. We have been talking about the basic structure and functions of cell, plasma membrane, and mitochondria. As we know, proteins and lipids are synthesized inside the cell. But do we know which cell organelle is involved in the synthesis of proteins and lipids? This cell organelle is endoplasmic reticulum or ER. The endoplasmic reticulum or ER is a network-like structure. As we can see in this figure, it is a network-like structure. And that's how the endoplasmic reticulum looks like. As we can see, it is a large network of flattened sac-like structures. These, the sac-like structures and tubules. So it basically consists of flattened sac-like structures and tubules. As we can see, it looks like a net-like structure or a network-like structure. For this reason, the endoplasmic reticulum is called as the reticulum because it looks like the network or a net. So now if we talk about the structure of endoplasmic reticulum, as we can see that the endoplasmic reticulum is in continuation with the outer membrane of the nucleus. This is nucleus and it is its outer membrane, and the endoplasmic reticulum is in continuation with the outer membrane of the nucleus. We can see these granules that are attached on the surface of endoplasmic reticulum. These granules are actually nothing. These are ribosomes. And as we all know that ribosomes are the site for protein synthesis, or ribosomes are the cell organelle which are involved in protein synthesis. So we can see that these ribosomes, number of ribosomes are attached on the surface of this endoplasmic reticulum. And for this reason, this endoplasmic reticulum appears rough because there are a number of ribosomes attached on its surface. So it looks rough, and for this reason, this endoplasmic reticulum is known as rough endoplasmic reticulum because it contains lots of ribosomes attached on its surface. But this endoplasmic reticulum, as we can see, there are no ribosomes attached on its surface. It appears smooth because there are no ribosomes attached to its surface. So this endoplasmic reticulum is known as smooth endoplasmic reticulum. And these flattened sac-like structures, these flattened sac-like structures are called as cisternae. So this is all about the basic structure of endoplasmic reticulum. As we can see, there are rough endoplasmic reticulum, smooth endoplasmic reticulum, and ribosomes and cisternae. So now we know that endoplasmic reticulum is a continuous system of cisternae and tubules, and it can be of two types, rough endoplasmic reticulum or smooth endoplasmic reticulum. Rough endoplasmic reticulum means that it appears rough because the ribosomes are attached on its surface. But smooth endoplasmic reticulum is smooth because there are no ribosomes on its surface. So now we will talk about RER or rough endoplasmic reticulum. As you can see, this is rough endoplasmic reticulum because there are ribosomes on its surface. So as we all know now that it appears rough due to the presence of ribosomes. Its membrane is continuous with the outer membrane of the nucleus, and these ribosomes which are attached on its surface are responsible for the synthesis of proteins. And in the ER or endoplasmic reticulum, proteins fold into their correct shape. Like proteins are being synthesized by ribosomes, but they are being folded by endoplasmic reticulum. Some proteins actually need to stay in the ER, but most of the proteins are then transported to the Golgi apparatus in the membrane vesicles. And these destinations, like uh, the transportation of proteins, synthesized proteins, can be to lysosomes, the plasma membrane, and the cell exterior, means outside the cell. It depends upon the requirement of the cell. This is actually how the proteins are transported outside the cell or cell exterior. So first of all, we can see that proteins synthesized by the ribosomes enter into the RER. So first of all, what happens? The proteins, as we all know, that proteins are being synthesized by the ribosomes, and these ribosomes are actually uh, transporting the proteins to RER, or rough endoplasmic reticulum. And there, in rough endoplasmic reticulum, the modification or folding of protein occurs. After this, when the proteins are properly folded and modified, these proteins are packed into transport vesicles. These are transport vesicles. These transport vesicles actually pinch off the RER, or rough endoplasmic reticulum. And then these transport vesicles come and fuse with Golgi apparatus. And then they are just transporting their enclosed protein to Golgi apparatus or Golgi complex. Now, the Golgi complex actually process these proteins or covers them, enclose them into vesicles known as Golgi vesicles. These are the Golgi vesicles, which are containing the protein, enclosed protein, and this protein, uh, these vesicles which are containing the enclosed protein, then pinch off the Golgi complex and then move towards the cell membrane or plasma membrane. Then these vesicles fuse with the plasma membrane and release their content or enclosed protein outside the cell. These are the proteins which are released outside the cells. So that's how the proteins are synthesized and then finally released outside the cell. Now, SER, or smooth endoplasmic reticulum, now we know that it appears smooth as no ribosomes are associated, but this SER, or smooth endoplasmic reticulum, is not involved in synthesis of proteins, but it is involved in the synthesis of lipids or fats. It also detoxifies the drugs and harmful chemicals in liver cells. So basically, SER, or smooth endoplasmic reticulum, is involved in the synthesis of lipids and detoxification of drugs and harmful substances. So these are a few basic functions of endoplasmic reticulum. They play a vital role in the synthesis of proteins and lipids. And now we know that RER, or rough endoplasmic reticulum, is involved in the synthesis of proteins, while the SER, or smooth endoplasmic reticulum, is involved in the synthesis of lipids. Endoplasmic reticulum, as we have already seen, transport the proteins and other biomolecules to cell organelles such as lysosomes, Golgi apparatus, plasma membrane, etc. So it actually uh, transports the substances to different destinations. So let's recall whatever we have done till now. Proteins and lipids are synthesized in endoplasmic reticulum inside the cell. Endoplasmic reticulum is a network of flattened sac-like structure and tubules. Endoplasmic reticulum can be of two types, RER and SER, rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum appears rough due to the presence of ribosomes on its surface. Ribosomes attached to RER are the site for protein synthesis. most of the synthesized proteins are transported to different destinations, while some of these proteins remain in the ER. SER appears smooth as no ribosomes are attached. SER is involved in the synthesis of lipids and detoxification of drugs and harmful chemicals. Thank you so much for watching. Please like, share and subscribe.